My name is Marla Schwarzfeld, and I'm a research scientist with Agriculture and Agri-Food Canada in Ottawa. And I work in the Canadian National Collection of Insects, Arachnids, and Nematodes, working on soil mites specifically, doing the taxonomy of soil mites, and also on soil biodiversity more generally. There's a whole world of tiny things that are living in the soil that you can't really see unless you get really up close and personal, and better yet, under a microscope. And so the groups that I work on specifically are mites, which are ridiculously diverse. And then there are also things like springtails, nematodes, other little tiny worms, centipedes and millipedes and all kinds of tiny things that are doing all the things underground that we expect animals to be doing above ground. And so there's predators and there's herbivores and there's things eating fungus and there's diggers and there's all kinds of different creatures doing all sorts of different things, just doing it on a really micro scale. For the organisms I study, the main sort of tool we use to get at this world is called a Berlesi funnel, or a Tolgren funnel is another name for it. And essentially, what we do is we take advantage of the animal's natural behavior of trying to avoid desiccation, which is that if the soil starts to dry out on the surface, they'll burrow deeper down searching for moisture. So the primary way that we get these organisms out of the soil to look at them is we take some soil, put it in a Berlesi funnel, which is just a metal funnel with a screen, and then we add a heat source on top, which gradually dries out the soil, and then the animals will come down, and then they'll eventually fall through the bottom and collect them in a collecting jar at the bottom. And we can do this live so that we can look at them, but most often we collect them into ethanol so that we can get them on a microscope after and take a look at them. They're hugely important for so many processes that we depend on, and essentially because they're processes that plants depend on. And so, everyone who's ever planted a bean, everyone who's tried to grow something knows that the soil fertility is really important, and these guys are huge contributors to soil biodiversity. So there are certain groups that's basically what they're doing is they're munching down on fungus and decaying matter, and they're pooping out these little pellets, and those pellets are really fertile, sort of nice crumbly soil that make things grow well. So they're contributing to soil fertility. They're also potentially eating pests or eating each other. So they're processing all the material in the soil and essentially creating healthy soil habitats that plants can then grow on. Basically, anytime you change the soil habitat, you're going to change what lives in there. For agriculture, one of the main issues is just a lot of these organisms are not used to living in this high disturbance environment. So if you go through and you plow your field every year, you're breaking up that soil structure, you're taking out, a lot of the nutrients, and then you're left with sort of finer soil without. So these things that live in the soil are living kind of in the spaces between the soil. There are other organisms that are living in water pockets, and then the mites are living in the air pockets, and they need that structural soil to live their lives. When you start breaking up that structure, then there's fewer places for them to live. Urban expansion is going to affect the soil fauna, because there's still huge amounts of diversity in cities, in the soil, but the more you sort of simplify the above ground habitat, the more you're probably going to be simplifying the below ground habitat. And so by losing all those different types of habitats, you're going to start losing the animals that live underneath. And one of the issues, of course, is that we know so little about what lives in the soil that we're not even, we know that we're having an effect, but we don't really know the magnitude of the effect or what kind of an effect we're having. I think I'm really a naturalist at heart, so I've always just been fascinated by biology and biodiversity particularly. I was a birder and I identify plants and I'm just interested in knowing what's out in the world. So I took a very circuitous route to become a mite taxonomist. My background is actually studying parasitic wasps, which I still find totally fascinating. And I still remember the first time I looked in a microscope and saw all these little tiny insects, these little shiny wasps, and I said, "These are amazing. I want to identify them." And I was told, "Well, you can't. Like they don't have names. There's no keys." And I just thought, well, this can't be. Like, someone's got to know what these things are. So that's what really led me into taxonomy and feeling like I needed to put names on things